Hi, everyone. Welcome to Being Patient Brain Talks. I'm Deborah Kahn, founder of Being Patient. Well, we all know about this perennial argument between beta-amyloid plaque versus tau tangles. What's the right target as we look for a cure for Alzheimer's disease? Today, we're going to talk about tau, more specifically, creating a tau vaccine in order to prevent and cure Alzheimer's. Joining me is Professor Norbert Žilka. He is joining us actually from Slovakia, our first guest from Slovakia. He's from AXON Neuroscience, which is developing this immunotherapy for tau. Welcome, Professor Žilka.
Thank you so much for this kind of invitation.
So, as I mentioned, a lot of debate among scientists such as yourself. A lot of people have been betting on beta-amyloid plaque as the target for Alzheimer's disease, but you have been studying tau for quite a period of time. Why do you believe tau is a better target than beta-amyloid?
I think that I should start directly with the history of our company, because it's nicely linked to the Laboratory of Molecular Biology, MRC Cambridge, UK, where our co-founder of the company, Professor Michael Novak, went just for 2 months. And finally, he spent there 8 years working together with 3 Nobel Prize laureates. And one of them was Cesar Milstein. And at the time, that was the year 1986, I think, they started to collaborate with Sir Martin Roth, who was a leading person in the psychiatry working on the Alzheimer's disease. And he had a kind of very good relationship with Aaron Klug, who was the director of the laboratory. And they wanted to identify the driver of neurofibrillary pathology in Alzheimer's disease. And Professor Novak was a member of the team, and together with the Cesar Milstein, who was awarded the Nobel Prize for the discovery of the hybridoma technology, which is an important approach for the producing monoclonal antibodies. So the main constituent of neurofibrillary tangles. So that's the reason why we started with tau. And then in 1999, Professor Novak returned to Slovakia after those 8 years and founded a company. And immediately we started to focus on the role of tau protein in Alzheimer's disease, but also in the physiological condition. So basically, that's the reason why we focus on tau protein.
Okay, I want to go into a little bit later how the vaccine would actually work. But first, I want to talk about, because it's endlessly debated in science, and I think it's fair to say more people have been in the past betting on beta-amyloid than tau. Now, the way that we understand the pathology of Alzheimer's is it starts with the plaques, and the plaques can be present in your brain for decades before you actually see a symptom of Alzheimer's. Now, tau comes, I believe, after the plaques. Is that correct? Is that fair to say?
I don't think so. Basically, this notion is based on the data coming from the biomarkers. So biomarkers in cerebrospinal fluid or PET imaging, but not from the post-mortem analysis. And we know that if we look at the very early studies, which did Professor Heiko Brack, we can see that tau pathology can be present also in a very, very young people, younger than 40. So at least we should say that tau and amyloid pathology is present more or less in the same time in the brain. Maybe it's only the question of the ability to measure the proper biomarker either in CSF or by using the tau PET imaging. And that's the reason why we can see this kind of delay between the tau pathology and amyloid. But based on the data from the postmortem analysis, we definitely can say that they started more or less in the same time.
So that's really interesting, and I have to be honest, you're the first person I've heard say this. So is it possible, can we presume, because there's people who have plaque in their brains and never see a symptom of Alzheimer's. Now, whether or not they've passed before they actually have the onset, the trigger of the disease, inflammation, or whatever it is it possible, do you think, to live with tau as well and never get Alzheimer's? Is that possible?
Yeah. Basically, it's possible. It's a very complicated process, the formation of neurofibril pathology, and it can take basically years. There are some studies showing that we are talking about 10, 20 years. There are lots of patients over 85 years having the typical pattern for Alzheimer's disease, and still they don't have any clinical presentation of dementia. So unfortunately, the tau pathology as such is most probably the part of our aging or aging of the brain. But there are some other factors that play important role, and that's definitely neuroinflammation, whether the brain is able to somehow cope with the presence of the pathology, tau pathology in the brain, or on the other side that it can somehow speed up that process. So we know that this could be one of the explanation why we can see some patient having lots of tau pathology and no clinical presentation, but usually after the age 85 years.
Yeah. So why, though, do you believe, I mean, if beta amyloid and tau are kind of appearing around the same time, presuming they are, and setting off, this process of neurodegeneration, why tau versus beta amyloid plaque? Why is it a better target?
That's a good question. I think that we should go back to the 1906 when Alois Alzheimer described the first case of Augusta D. And he analyzed her brain, showing that both structure, amyloid beta, senile plaques, and tau neurofibril tangles are characterized the Alzheimer's disease. And since then, we accepted this view on the disease because there are many other forms of dementia. And in some of them, only tau alone is a driver. But in that case, we can see a slightly different clinical presentation. So in other words, tau can do that job independently from amyloid. But in Alzheimer's disease, most probably these two pathological pathways somehow collaborate and create the specific and typical feature, which we call Alzheimer's disease.
So we have a question that's come in saying scientists are still trying to figure out the basic functions of beta amyloid. I mean, there's also hypotheses that, maybe some plaque is okay. It's part of the process, and it's when you reach the tipping point and things go really wrong, then you set into neurodegeneration. But so, they're still trying to figure out the basic functions of beta amyloid. What's the role of tau in healthy brains? Is there a role for tau or in healthy brains?
Of course, yeah. Maybe I should start with the basic information. The tau is peculiar protein because it belongs to the group of proteins which we call naturally disordered protein. And it means that these proteins usually do not have the final structure. So imagine like spaghetti in boiled water, that they can change the structure, let's say, every second. So that's the typical feature of physiological tau protein. And that's a great advantage because then it can serve as a signaling molecule. And there is one important feature that they can attain the specific structure upon the interaction with other proteins. And we know that tau protein can be distributed throughout the neurons in physiological condition. So we can analyze the presence of tau also in the extracellular space. Of course, tau play important role in the synaptic functions. It's important for the cytoskeleton of neurons. We can find also tau in the nucleus of the neurons, but not only neurons, but also other types of glial cells. And in nucleus, they can somehow protect DNA. So there are lots of roles for tau protein, and this is due to the fact that it has no firm structure. So tau protein is very flexible and can do multiple jobs.
So it's not until it really reaches a tangled state, right, where things go wrong. So, yeah. So we all have tau, but when that process becomes destructive is when you're actually seeing the tangles. Mm-hmm. So you're developing a vaccine. It's been, you just finished clinical two trials. Mm-hmm. Tell us exactly how the vaccine works. I mean, you're only targeting tau tangles, I presume. So is that enough to really stop neurodegeneration?
Okay, maybe I just want to a little bit elaborate on the main difference between the physiological and pathological tau, because it's important for the description of the function of the vaccine. So first of all, I should say that when we are talking about Alzheimer's disease, we usually say that the tau protein and amyloid protein represent the main feature of the disease. But we should rather say the tau proteins. We need to use a plural in case of tau, because despite of the fact that there is only one single gene for the tau protein, during the some production, we can easily find 6 different isoforms. In normal physiological condition, 6 different tau proteins with a different structure and different function. But on the top of that, there are multiple modifications. Again, in physiological condition, and again, you can somehow modify the functionality of that particular tau protein. So in normal brain, we are talking about, let's say, maybe hundreds of different tau proteins. Different structure, different function. And in Alzheimer's disease, it's even more complicated because there are much more we call that post-translation modifications. So you can get the huge family of tau proteins. And in case of Alzheimer's disease, we can talk about the maybe several thousands of different tau proteins. So what's the main difference between these group of the physiological and pathological one? The main difference is in one very specific modification, which we call truncation. Truncation is normal physiological process. You can find lots of truncated tau protein in the physiological condition. But that kind of truncation is completely different than truncation which we can observe in Alzheimer's disease brain. So basically, you can get the different types of truncated tau protein. On one side, normal truncated tau proteins. On the other side, the pathological one. So unfortunately, brain is not able to discriminate between bad and good guys. There is no specific information. But immune system can do this job. But for that, immune system needs a specific information. And this is all about our vaccine. So the approach was to find whether the immune system can somehow find the way how to differentiate between these two groups. We are still talking about more or less the same proteins. The primary structure, the sequence is similar or the same between the normal and pathological tau proteins. So how can immune system select it between these two groups? And we found that there is one main difference, and the difference is based on the conformation. So in case of pathological tau species, let's say, we can see that there is a specific conformation, specific shape of those truncated tau proteins. And the antibodies can nicely recognize this type of structure. And because it's different between normal and pathological, these antibodies usually do not recognize the physiological tau proteins. So what we did was that we identified on the tau protein the specific short area, which we call Achilles heel. It's a vulnerable area on tau protein, which is responsible for the pathological tau-tau interaction. That's the reason why we can see neurofibular pathology. And we use this short part derived from tau protein as a main component of the vaccine. Of course, that was very short peptide, and unfortunately, it's not immunogenic, so we are not able to stimulate the immune response. So for that purpose, we use a carrier protein. So we coupled this short tau peptide to the carrier protein in order to persuade the immune system to produce the specific antibodies against this very small area on the tau. And interestingly, the same area you can find in both groups, in normal tau proteins and in pathological tau proteins, but the structure is different. So,
right.
Yeah.
So just tell us a little bit about how this trial. So, phase 2, you were giving a smaller group of people it's an injection? What is it that you're administering? I mean, immunotherapies are drugs that are given to us to boost an immunoresponse to something, which is what you just described. Yeah. So how do you administer this? Is it a one-time shot like a flu vaccine, or is it something where you need to get infusions every couple of weeks? Yeah, so there are two main approaches, immunotherapeutic approaches, which are widely used in the clinical trials these days, either for amyloid or tau. Mostly the pharma companies are using the monoclonal antibodies, so that's already final version of the antibodies, and they can inject those antibodies intravenously once per month. In our case, the vaccine generates these antibodies in the body or bodies of the patients, and we apply the vaccine subcutaneously, like every other vaccine. And because we are talking about the therapeutic vaccine, not preventive, in that case, we need to apply several doses. In our case, the regime was based on the 6 initial doses, 1 per month, and then every 3rd month we applied one as we call that booster, just to keep the immune response in the same level. So what were the results of phase 2 to warrant phase 3 trials? Okay, so one of the most important results coming from the phase 2 clinical trial was that the vaccine is safe. And that was very important because we recruited elderly patients older than 55 years. So we didn't find any serious adverse events during this clinical trial. Then what was the second main point was that the vaccine was strongly immunogenic, which is not easy for in general for vaccine, because majority, if not all, people over 50 suffered from so-called immunosenescence. So they respond not properly to the different type of vaccines. So that's the reason why we somehow modified the vaccine in order to get the proper immune response in these elderly patients. Were you able to see that it actually worked in terms of, setting off this immunoresponse aimed at the bad part of tau, to put it in layman's term.
So the third point, which is important in terms of the phase 3 or the further clinical development, is that we were able to see that the vaccination slowed down the neurodegeneration. We used several biomarkers for that purpose, and we found a statistically significant effect on the neurofilament in the plasma, which is widely used as a biomarker of neurodegeneration in human brain. And we also observed the effect on several tau biomarkers in cerebrospinal fluid. And when we selected the patients having the typical amyloid and tau pattern based on that predictive model developed in University of California, San Francisco, we found also the effect on the cognition and some functional assay. So basically, we can say that the vaccine could slow down the neurodegeneration. And if we properly select the patient in the next clinical trial, it means that those patients will have both amyloid and tau pathology. I think that we will be able to somehow replicate or confirm the data from the phase 2 clinical trial.
So is it that this vaccine, when you're picking people in early stage Alzheimer's, they've obviously started to go into neurodegeneration. So I'm presuming the vaccine can't reverse anything, but rather it could prevent more degeneration. Is that correct?
Yeah, I think that's correct for every therapeutic approach, because we need to be aware that when the first clinical presentation usually appear, at about 40–50% of neurons are died in special brain areas. So I think that the main limitation is that there is no pain in the beginning, and so the many patients in the early stages somehow ignore the first clinical symptoms, and they come too late to clinicians. So that's the reason why we would like to focus on early stages, hopefully on MCI.
Sorry, I don't mean to interrupt, but could this be used as a vaccine to prevent? So if I don't have any signs, could I just get this vaccine even though I'm not on the road to Alzheimer's, or at least I don't know I am, but I may be, but nothing obvious. But could I get if the trial turns out that it is effective, is this something everybody could use, whether or not you know that you're on the road to memory loss?
Exactly. This is the ultimate goal of our company, that once we will be able to identify the therapeutic effect, then we would like to focus on the prevention. In that case, there are several approaches which we can use for that purpose. One of that is that we can go for people, still healthy people, healthy volunteers, having already amyloid tau biomarker positivity, either in CSF or by using the PET imaging. So this is the special vulnerable group where we can expect that maybe in future they can develop dementia. So maybe this could be the next step in our clinical development.
So I guess the question that probably everybody wants to know is, how far away are we from knowing that this vaccine is actually going to work? I mean, we spoke earlier, COVID certainly isn't helping things right now, but how far away, when do you expect you'll know, and what will it take for you to understand that indeed this could be an actual Alzheimer's treatment and prevention?
Basically, we wish to start as soon as possible, but due to the COVID situation, I think that realistic date is most probably next year. So next year we can start the latest phase of the clinical development, the phase 3, and then we need to wait at about 4 or 5 years until the final conclusion.
And what about treating things like CTE? You know, the a lot of athletes. Would this work?
Of course, this is one of our next step, I should say, because we know that especially in CTE, tau pathology plays an important role. It's a kind of reverse Alzheimer's disease, where the tau pathology first appears in the frontal cortex, frontal part of the brain, and later on the hippocampus and entorhinal cortex. And we identified the presence of these kind of truncated tau proteins, the BET truncated tau proteins, also in the brain of CT patients. So the target for the therapy is there. So hopefully we will be able to focus in the next clinical development also on the CT patients.
Okay, well, thank you so much, Professor Norbert Žilka, for joining us. This is really interesting research. We'd love to stay in the loop as you discover more. We wish you all the luck. I mean, if it could be as easy as getting a vaccine, that would be so life-changing for so many people. So thank you for your research. Please keep us abreast of any of the latest developments. It's a topic, you know, we often talk about. So thanks so much for joining us.
Thank you so much for the invitation.
So as always, if you want to see these interviews in their entirety, go to beingpatient.com. Please sign up for our newsletter. We keep you where we keep you notified of upcoming talks. We have scientists from all over the world joining us on different aspects of Alzheimer's disease, brain health, treatment, the search for a cure. So please join us, and thanks so much for watching.